Hey guys, thank you for sharing your excitement for science or technology in particular in those interviews I could make with you on the last day of the exhibition. Hey, could you tell me a few sentences what is special about your research?
So I researched two species of bee pests, the wax moths, that might be able to degrade polyethylene. They're like really basic plastic that we use everywhere. So if this was true, we could maybe help save the environment with it.
That's great. Congrats on your research, and I'm just gonna take a closer look.
Yeah.
Like these are the experiments you've done, pea samples. So you were working with larvae. What was it like?
It was disgusting mostly, but it was interesting.
Okay. In conclusion. Oh, you were also done the references. Smart. Yeah. You can be scientific. Okay, thank you.
My project is a new way that we can use oxygen-based hyperbaric therapy to treat neurodegenerative disease like Alzheimer's disease and Parkinson's disease that are related to DNA damage. And this project, its major implication is that it shows us new kinds of neuropathological conditions that we can use oxygen-based therapy to treat.
Okay. Can I get...
This is a protein called 53BP1 that indicates DNA damage, and you can see those red dots. This is accumulation of 53BP1 in the damaged site in the nuclei. So when there is damage to the DNA, there is accumulation of the protein, and it appears like red dots in the nuclei.
I see. And these are the astrocytes?
These are astrocytes, yes. And here in green you can see GFAP, which is a glial fibrillary acidic protein. And it is a protein that indicates stress. When astrocyte is under stress, then it expresses this protein. And when you quantify it, you can compare the study group and the control group and see that cells that were exposed to more oxygen were less under stress.
Okay, that's great. I'm taking a look at everything and just getting a video. That's cool. Thank you for explaining it.
So we were concerned with one of the most important problems from the last century of mathematics, and we took a computer to formally verify it. That is, we encoded the problem in a computer and in the language of the system Isabel, and we made sure that every single step is correct, doesn't have any errors, and is self-consistent.
Okay, cool. I'm gonna take a closer look on the posters. So this is the timeline, how the project developed throughout the years. Okay. I get it. I'm gonna take both of them. Okay, Isabel started 1986. Oh, this you and your teammates.
Exactly.
I thought it's like an album cover.
This is a picture from our national competition. And then the gag we always do for the jury is once we finished our project, then Hilbert will have met Isabel. Oops.
Thank you for explaining.
Thank you very much.
So I'm from Luxembourg, and
yeah, I see your project.
Yes, and my project is about L-carnitine, which is a protein, and not many people know about it, but it actually has an effect on fat metabolism in the body. So in a way, it helps fat loss and weight loss, and the build of muscle. So what I did was I used larvae to test this, and at the end I found out that it does really have an effect on losing fat, building muscle growth, and metabolism in larvae.
Okay.
So, however, too much of it can be harmful. So I think if I have the opportunity to, I would take my project further and maybe commercialize the use of L-carnitine and spread the word as much as possible.
Get well with the larvae.
It was pretty interesting. I was just like walking around with them for 2 weeks, and my friends and family weren't exactly happy, but you know, for science.
I can imagine.
Yeah.
Like you gotta do it for the sake of science.
And, this is the respirometer, and this is what I used in my project to measure the rate of respiration and how much the oxygen intake.
So for my project, I assessed the antiretroviral potential of 8 medicinal plant tinctures. And by that I mean I wanted to know whether they could have some clues for a new anti-HIV drugs. And I did my experiments in vitro in cell culture. The experiment, you can see a tincture here, 0.4 concentration here, and you can compare it with an official drug.
Okay.
Although you can't say right now that a tincture has an effect on an HIV infected.
Okay.
You would have to study that further.
All right. So you are planning to further your research in virology in the future?
Yes, I'm planning to extract the single compounds that had the effect.
Okay. And then use it in medical purposes to
Perhaps
treat.
if everything goes well, but the process is really long.
Yes. I get the struggle. Yeah. Thank you.
Sure.
It's very nice.
My project is about Ulam spiral, also known as prime spiral. Actually, it was made by Stanislav Ulam in 1963 while he was listening a very long and boring article in a scientific conference. And for example, it's also known as prime spiral because if you put 17 at the center, the diagonal gives you a prime full of prime numbers. There are numbers up to 272. And for example, if you put 41 at the center, it also gives you the diagonal is full of prime numbers. And I was impressed by prime numbers and life of mathematicians and their powerful intuitions. So I worked on Ulam spiral. Ulam spiral, and I separated a square to 4 different regions to find lower left corner of any given number in Ulam spiral. And these regions are valid for given numbers position.
And you've written the mathematical formulations yourself for the rules that you found in there.
Yeah, and also I found summation of the diagonals in given number squares, and like I translated Ulam spiral to 3-dimensional space, and I made cube spiral, and I used it in cryptology.
Wow, that's really impressive. I think these are the 3D pictures?
Yes. This is 3-unit cube, and for example, this is 4-unit cube.
Çok teşekkür ederim.
Rica ederim.
My project is about plants and how they affect bacteria and how they will be able, hopefully, to treat people in the future.
That's great. Can I take a closer look?
Of course.
Okay. Thanks. So you've extracted several solutions from the berry and you've tested it on bacteria. If I understand correctly. And here are your results.
Yeah, exactly. These are
Great.
different commercially produced samples.
Yeah.
And then these are also tested against bacteria, whereas the wild sample is a lot stronger. And
Yes.
this is a lot weaker. So.
All right. And they have higher content of phytochemicals
Precisely.
inside them. Congratulations and thank you for contributing to the blog.
Oh, hi.
Hello, can you explain more about your research?
Yeah, of course. So what I was studying was the effects of ethinyl estradiol on plants. This is the hormone we use in oral contraception, or we also give it to animals like that we grow for food. And what happens with this hormone is that it's excreted by our bodies and also by animal bodies, and then it gets to the environment. And since we use this water and manure as fertilizers and water for irrigation, this may be very harmful for the environment and for the whole ecosystem, and also for the plants we're trying to grow. So what I found out is that if this hormone comes in too big concentrations, mostly from the fertilizers, it may cause the plant growth to be inhibited. So we should pay attention to these things.
You drew these?
Yes.
It's great. Okay, nutrients and this is the hormone?
Yeah, it's the hormone.
Okay. 2 and the petri dishes?
Yeah, it was I also grew fungi, so that's I grew. Thank you.
And I hope the best of luck to you.
Thank you. Bye bye.
Bye.